So I wanted to talk a bit about genome reference structures, the way that we sort of interact with the genome. You've heard other talks from people involved in such things. I think now is actually an interesting time where we are going through certainly an exploration of alternative ways of representing reference information for genomes. And maybe we're going to enter a new paradigm. You never know until it's happened, so we'll see. But OK. So we all rely on reference genomes. And some of you will have heard some of this talk before. I hope none of you have heard all of it. I don't think that would be the case. There's new things in here. But obviously, there's an accumulation of different kind of threads of argument about, or of ideas about reference genome structures. So we rely on them for mapping shotgun reads, and that's the standard paradigm for sequencing. Is you make a library and you shotgun, which is made of fragments that may have come from lots of different sources. It's a very powerful approach, actually introduced right at the beginning by Fred Sanger in the 1970s, amazingly, and some of the very first genome sequences, or first sequences, were assembled that way with the Staden package, so in the prehistory of genomic bioinformatics. But in order then to work out where they come from, you have to have a map, which you can put them in. And so we use reference genes for mapping sequencing reads. And that immediately raises an issue that mismatches are either errors, which you're not interested in, or they're genetic variants, which possibly is the thing that you're really looking for. And you have to distinguish between things you absolutely want to throw away and things that you really care about. You can also have mapping errors where you don't actually get right where the read sits. And those can be either global or local. So they can be global, this really belongs somewhere entirely different, or local in the sense that you actually have identified correctly the right place in the genome, but you're not quite sure how it aligns. And all of these sorts of errors can also lead to misinterpretation. So that's a key function that's non-trivial. And the second important function is to provide a coordinate system for reporting and annotation, for tying together information. And I remember actually around the time of the Human Genome Project, 15 years ago or so, talking about building a reference coordinate system onto which people could layer, in fact, in the context of Ensemble, could layer additional information, and how that would be good to do with maps, real maps. And a couple of years later, and actually the UK Ordnance Survey should make its maps fully public, and then people could layer extra information on them. And a few years later, as the internet developed, Google introduced Google Maps, which do exactly that. And that function of layering things on is really important. So there are genome browsers, and bringing together things on a coordinate system is a key function of a reference. But as well as the reference genome being a sequence, reference is really all that you know, well represented, that you can use in other ways. There are resources of known variation. So, for example, dbSNP, RefVar, the 1000 Genomes Project dataset, and so on. So those form part of the reference genome. So, we have a solution that we're all using. Why should we consider look at this again? Well, it is a bit messy around the edges. There are issues around these things. We do pretty well for most of the genome. We're sort of in the 95% range, maybe. But sequence data acquisition is continuing to increase, and I think will continue to increase. We're still, it's not yet done, the genome revolution. We're still gaining more information. Even if prices don't continue to drop, and we hope that they do, and there are all sorts of new companies, the amount of data being accumulated is increasing, and we can't stand still, I think. And ultimately, we want fast and accurate methods, and we want to apply them to compressed data, and I think that's going to become more and more important, is ways that we can use compressed data and use it at the same time effectively. And some of this has come in with the sort of CRAM and new sorts of versions of BAM files and other sorts of compression, but I think that's going to be an important trend going forwards. So how can we hope to carry on getting better? This is faster than Moore's law. So we have to become algorithmically smarter. And I think generally the field represented in this room has done pretty well, actually, over the last 20 years in getting algorithmically smarter on a sort of 5-year time frame so as to keep up with this sequencing technology. And the key fact is that we're related by descent from a common ancestor. The sequences are not all novel. If we got new things every time, life would be very hard. The more people that we sequence for humans, the more we keep finding variation we've seen before. It's a bit more messy in metagenomics, but there, again, increasingly, as you sequence more and more samples, particularly from the same sorts of samples, you're getting similar sets of information, and you want to represent. And so you can relate what you've seen to what you've seen before, and increasing, the more you get, the more like something you've seen before it is. And we have to exploit that. But when we do so, there are going to be trade-offs between the complexity of the reference and the degree to which other new things seem novel. So if we build in all the things we've ever seen before into the reference, then it's going to be a very complicated thing. But very little of what we see, again, in the new sample will be novel. If we have a simple reference, then we can understand it. It's a kind of linear thing, but more of what we see will be novel. It's that trade-off that we sit in. I think we should recognize that. And I think in time we have to be prepared to make the reference more complex so as to manage new data in a graceful way. And we have to provide the sort of toolkits as a field to support all the things that people want to do and the interpretation. So that's one issue. The second issue is, although there is this trade-off, we don't want to bias too much towards what we've seen before. And ultimately, you could say what you'd like to do with any new data set is sort of start from scratch again, because that's unbiased. So there's some trade-offs here, and one just has to always be aware of those. It probably doesn't make sense to completely ignore everything we've known before, but at what point to bring in previous information and how to relate that to a sample are key ideas. Okay, so right, there's a clock up there, good. Okay, so I'm going to start. I'm not actually going to talk a lot about GRC. Other people in the audience, I think, have talked and will talk about the Genome Reference Consortium. We're involved in it at Sanger Institute, along with NCBI and WashU and EBI. The GRC supports the standard human reference and develops it and keeps it active and relevant. And in December 2013, over 2.5 years ago, they released GRCH38. It's much better in all ways than GRCH37. And the field as a whole have not really moved completely to it, but I think that's happening. And I wanted to point out, there's over 7,000 variants fixed, or errors fixed. There were, for mapping purposes, the 1000 Genomes Project found it made a big difference to add a whole lot of sort of adjunct decoy sequence to help avoid biases in mapping to GRCH37. And most of that, 3/4 of that is represented, and the rest is kind of now managed and coordinated within the GRC framework, so you can get from GRC an entire package. So we've dealt with most of the problems that was introduced to evaluate. As people here know, there are these alt loci. They're important because they're regions that are so divergent that you can't map easily to a single sequence. Most are small, some are large. And the VWA, which is at least one of the core, sort of paradigmatic mapping algorithms, now maps totally happily to a reference, including those. And I wanted to say, so Hung Lee introduced in BWA-MEM, the current version, the capability to handle GRCH38 with alts over a year ago, maybe 2 years ago. You can then use standard calling processes. What happens is that things that map equally well to the primary and the alt don't get penalized. They map to the primary. If they map better to the alt and they're not representing the primary, then they'll get placed on the alt. So you can make variant calls on the alt only in those regions which are distinctive from the primary. And it substantially increases mapping performance and reduces false positives. And I hope we're turning the corner now that people are beginning to do things this way, because actually, as I say, so I want to send the message it's better. So I heard TopMed, the kind of big consortium in the US of whole genome sequencing, have switched to it. Sanger has switched to it some time ago for new whole genome human sequencing. And the 1,000 genomes has all been remapped to it. Okay, so I want to move on from there. So can we go beyond the alts? Why do we keep on? If we map even to GLCH38, a new individual, or data from a new individual, over 99% of what we find we'll have seen before somewhere, and it'll be represented already in reference databases of variation. And that's inefficient, and it's not only 99% of the novel, or of the things that are different from the reference, which are true, it's also, actually I don't know this number for sure, certainly, but I would suspect that something like 99% of the errors we'll have seen before as errors. And at the moment, we're not really dealing with that. And we have all sorts of filters in place that we apply from scratch to try and filter the errors out. And some people have blacklists of things, but ultimately, why are we ignoring that information? So there are two ways I'm going to talk about of going beyond, of building in some variation information into the reference so that we can take account of it up front. So one is to use a graph reference using local variation and map to a structure that includes that. So allowing you to know that there's a SNP which is quite likely present in this location. But the second way is long-range variation, haplotype structure. So that's saying that because we've inherited the sequence that we get from a new person will be related to somebody else who has been sequenced before more closely than they are to the reference genome. And they'll share their genome ancestry in pieces, which can be megabases long. And you can use that information essentially because 99% of the recombination breakpoints that you see in a new person relative to the reference will be seen in, would have been seen in other people before. And this is because there's actually, if you look at the basic mutation rate, which is about 10 to the minus 8 per base pair per generation, is about the same as the recombination rate, 10 to the minus 8 per base pair per recombination. So there's really a dual sort of structure of generates variation in genome sequences. And we shouldn't just think about the mutational side. We should think about the recombinational side when we're trying to relate what we've seen to our previous knowledge. So I'm going to talk a little bit about that as well. For people who were here in the workshop that the GRC ran on Monday morning, I talked about this. There's some overlap, but I've also changed the things I say here a bit as well. Okay, so I'm going to start by talking about the variation reference in terms of about the graph reference approaches, the graph structures. So the idea here is that rather than have a single sequence, which we use as our reference genome, a variation graph represents many genomes in one non-redundant structure. I keep on clicking this, and that's because I'm using it too much. So basically, you can follow along a path, and where it's known that there are two different alternates, you can pick either of those. And so this thing here is a sort of fairly condensed way of representing a whole set of combinatorially different set of sequences. And that's a sort of exaggerated version. Most of these will be SNPs, actually. But you can have more complicated structures like this, where they're indels, like the deletion at the bottom would delete that whole middle segment. So it's a cartoon. So this sort of idea people have thought about in some form or other actually for quite a long time. And there have been papers, particularly from Zam Iqbal and the group in Oxford and others on these structures. And the idea here is that nodes contain sequence in a graph, and edges between the ends of nodes represent potential links between successive sequences, and you can build up a sequence in that way. And because genomes are DNA sequences is sort of this skew-symmetric, bidirectional So as well as reading in one direction, you can read the reverse complement in the other direction, and those are biologically both functional. There's a little bit of extra sort of complexity to the graph and to mapping. We have to, as well as allowing GGCCCTT, you have to allow in reverse AAGGGCC. And that's actually the same as a train track. You know, a train can move in either direction. The two rails are actually different. You can turn it around and move either way. You can join things up so that you can get loops and cycles where you go around, or you can get inversions and all sorts of different structures. And so a good way, actually, to think about the different structures is to buy yourself a Brio train set and play with it to see whether things can join up appropriately. Okay. There are actually other ways to build these sorts of structures, but there's some intricacies. And there is actually a branch of mathematical graph theory, it turns out, called skew-symmetric graph theory, which deals essentially with these objects, surprisingly. I don't know if they were aware that they were biologically relevant. So, so as I said, there have been people using these sorts of structures before, but there's always been a problem that when you build graph structures in computers, you tend to need quite a lot of storage for nodes and edges and links and pointers and so on, and they're unwieldy for human genome-sized data sets. And I think that's been one of the barriers to adopting this type of approach as a reference. We also then need to have all the machinery of mapping reads of mapping data, of holding annotations, of visualization, and so on. So the VG project, which was sort of initiated, and key guy is Eric Garrison, who was involved in the 1,000 Genomes Project in Boston and is now at Sanger working in our group, but there are many others involved. So VG team is the collection. They are aiming to provide that infrastructure package, the software package, to allow you to handle at gigabase scale reference graphs. And it stores nodes with sequences, edges, paths, in particular mappings. So paths are routes through particular instantiations. And mappings are the things that allow you to say how another sequence relates to it, either a read or a cDNA or some version of the reference genome or so on. And there are alignment tools, and there's a version of the BAM format, .gam format. And you can store it to disk, and there's a succinct representation, which is very highly memory efficient. You can hold in a sort of simple server standard, and tools for building and editing and extracting and unrolling and doing all sorts of graph operations. So this is really meant to be a toolkit for enabling us to work with graph genomes. And actually, this is a paper that I wasn't involved in. It's in press in briefings in bioinformatics. It came out a meeting on pangenomes, sort of mini microbial. But it has all these pictures of things that you'd like to do, and you probably can't read all these. This is taking read sets and aligning them, variant calling, phasing, updating, retrieving those things about individuals. This is building them from references and lots of alternate versions and constructing them, storing, retrieving them, comparing them, annotating, simulating, and visualizing. So all of those. And VG, as a package, has some form of software for, you can see here, for doing many of these jobs, essentially all of these jobs, possibly not the comparison yet. So it's a work in progress. I think it's probably not out there ready for a major project to sort of suddenly switch to using, but we hope to get it there in the coming year. And actually, a lot of it is usable, and certainly if you have smaller data sets, I think it's really worth exploring. And maybe there are things that that people see as missing, participate in the GitHub thing, but they're very open to having extra pieces put in. So one key thing is mapping. I think that's actually very central in the modern world. Our original approach was to use k-mers. Here's a read symbolically to try and do k-mer matches. This might be a bit of a graph running top to bottom with various variants in it. To cluster the hits and then build a sort of subgraph and do a sequence to graph alignment, of which there are algorithms dating back to the 80s, and store the results. But some of this became quite heavy, and I think you heard a talk, this is out of place. let's jump on. You had a talk by Uni Siren yesterday about GCSA2, which is the BWT version of the algorithm. It's the graph version of the BWT algorithms to map. And he's now got that working, so it effectively gives the same information. These maximal exact matches of reads or sequences into a graph, and we can use those as seeds for extension. So I think we've kind of conquered, in principle, the speed issues or Uni has conquered those for making this efficient, and the index for that stays under 20 gigs, can easily sit in a server. So I think not all the pieces to sort of the BWA and functionality are there yet, but there's versions of all the extension and read pair and so on algorithms, and so we're essentially there for an efficient mapping strategy. Okay, so that's what this was meant to say, but doesn't. As part of the Global Alliance for Genomics and Health, which I'll mention a little bit more at the end, Benedict Payton and collaborators sort of centered at UC Santa Cruz have been evaluating different graph genomes, saying what extra does this really give us in the human genome? On pilot regions, so they picked a set of pilot regions. These are the LRC, the MHC, the KIR region are particularly known to be nasty, have lots of alts in the current GRC reference. These are important medically and because they're parts of challenges for the Global Alliance. And these are all different graphs, some from academic groups, Santa Cruz, some from Severn Bridges also had one, Sanger. And then we built several of our own, including some controls. And we evaluated, how does it differ incorporating all this extra variation? And these are slides from Benedict, and they are probably draft, I should say, but I hope close to being draft. So this is in the MHC. This is the fraction. We're taking all the reads from the 1,000 Genomes Project from a variety of individuals. And we're asking what's the fraction which are uniquely mapped, and what's the fraction that are perfectly mapped. And it's not surprising. If you incorporate known variation, you can map more things exactly, which is this way, and we can get quite substantial, in this area that's very variable, improvements with some of these graphs. So this is the primary reference sequence. This is the fraction uniquely mapped. It's not so clear, but actually this is the significant increases here in the ability to be certain where something belongs, to disambiguate. There can be trade-offs. If you build in too much variation, then actually you can make it, there are trade-offs here. So that's the MHC. BRCA2, it's all, the sort of proportion uniquely mapped is much higher to start with, and the gains are smaller, but they're still in the right direction, at least for these graphs. The 1,000 genomes is right up here. You could say it's cheating because that's the graph built out of all the variation called on the 1,000 Genomes Project. And we're using 1,000 genomes reads as our test, and so that's not a totally fair comparison. But some of these are built in other ways. And we're still improving the perfect matching, as you might expect, because known variation is being incorporated. OK, so that's looking like it's working. What about genotype calling? So actually, the Santa Cruz group have been the key group pushing that. And there's a whole set of issues that happen. What actually is a variant in a graph version? Well, a lot of the variants, as we said, 99% of what you expect to see in a new sequence are within the reference. So what you're doing is really picking out which route to take, rather than necessarily calling a variant. But the GAM format that I talked about does allows you to have an edit string, a cigar string that says, I've seen something novel here. And if you have a lot of reads that do that, the strategy is that we build, just as in GATK, if there's sufficient read support for some variant, then you consider that. And the way we consider it is by temporarily adding it into the graph, using the graph editing functions of VG to add it into the graph, and then remapping the reads onto it so that we essentially deal with everything as a mapping thing. And then we have to define what a locus is and what alleles are. And so we look for bubbles, places where things diverge and come back together. So there's a bubble, there's one, and here's one. And they call those a locus, and then an allele is a path through that. And so that generalizes. It means indels look pretty much identical to, from this graph point of view, to SNPs. And you can deal with some of those more complicated overlapping structures, allowing more alleles. Okay, so you calculate likelihoods then for mapping and work in the same sort of way as before. We evaluated by taking the two PacBio haploid human assemblies that WashU with collaborators generated CHM1. These are from hydrotidiform mole clones. Taking Illumina reads also from those same samples and mapping them back to mapping into the graph, and then comparing those to a truth set, which was obtained by taking the assembly and mapping to the graph. And we mixed reads from two so that it was a pseudo-diploid. So the truth set was from PacBio. And it's interesting, we found that that was a better way to evaluate these things than if you take, say, the genome in a bottle or Illumina Platinum Genomes truth sets, which are basically built by using Illumina reads against the reference, and so are quite strongly biased to the sorts of things that you can record well that way. So we think this is a more neutral way to evaluate. And this is the F1 score, the sort of combined false positive negative. And you can do better with different graphs. Some of the free Bayes and platypus and so on do quite well, but we think we're also improving variant calling, and we don't think we're at the end of this. And one of the problems always in introducing something new is there's been a huge amount of effort in the existing technology, and introducing something new is always, there's been a lot of engineering that you have to go beyond. So here we're seeing different versions of the graph genome. This is the primary on black. This is a distribution of indels found. And we're showing that if you incorporate more variant paths in your graph genome, you can find more. So this is on a log scale. You can find more variants, more large variants. It's not a surprise if you start allowing building in known indel variation into the reference. It's easier to map to with short reads. And so we hope that, of course, you have to discover it to start with, but we hope that one can build and capture at least genotype better, essentially, from sequencing data, indels with this approach. Okay. And the final slide, which also I showed on Monday, says, what about the coordinate system and visualization? It is a struggle visualizing a graph. One thing you can do is for an individual, I think there have been talks about this, try and work out what the paths are and visualize that. And then you have to keep a translation of how the coordinates map between the reference and this new sequence which you're using. There's a whole machinery inside VG to maintain translations between versions. And one nice thing about this, which I feel is, we probably don't have to give up our GRCH38 coordinates. We can incorporate that into the graph, and maybe even 37, so that we can sort of visualize our results against either coordinate set and translate them. We do have to have a bit more machinery for handling that. Okay. Okay, so that's those are variation graphs on the local scale. What about this long-range scale I talked about at the beginning? This is where I was meant to show this slide that so I'm moving on to that section. So essentially, the way that we currently typically represent long-range variation is as a set of sequences. The HapMap2 panel 10 years ago had 210 people originally from 3 populations, 420 haplotypes since we're diploid, and about 2 million variants across the human genome. And progressively over the last 10 years, we've got a series of bigger reference panels. So HapMap3 was much bigger, 2,000 haplotypes. The 1000 Genomes Project started being sequence-based rather than genotype-based, and so got into the tens of millions of variants. So phase 3, 88 million variants. some of the new sequencing projects. So these are all fully public resources that anyone can use and download and build. There are bigger data sets, but essentially those are all under data access control, and humans, there are good consent reasons for not making them fully public. And there are hundreds of millions of variants known. So over the last year or two, we built with a consortium of people, including Gonzalo Abicassis' group at Michigan, and Jonathan Marchini and his team at Oxford, and a lot of collaborators who had projects. A reference panel of 65,000 haplotypes from 15 populations. They're mostly European, but we are planning to extend that across 40 million variants. So this is a really big increase in the number of haplotypes. And what do we use these for? So we use them for, the main use to date has been for imputation into genome-wide association studies. So here's an example of a genome-wide association study. The black dots were the genotyped variants, but there are additional sequence variants in those samples that weren't genotyped. And if we have a reference panel that incorporates them, we can infer what the sequence at those missing places would be. And we can get more strong signals. This is at IL-23R, which I can read. And basically, we can get extra information. And this has been the key method for genome-wide association studies over the last few years to both improve power, but in particular to integrate data from different sources and look at variants that weren't genotyped. So meta-analysis on rare variants. Also, that we can use this for statistical phasing of new samples. We can take a diploid and try and tease apart the haplotypes using known haplotype structure. This is the same technology that underlies the low-coverage sequencing, which Daniel referred to, so that we're sharing data between individuals based on haplotype matching, on sequence matching. And it basically is capturing the variation amongst populations along with the point variation. It's an important resource for understanding human genetic variation. So the HRC, we built out of all these different projects. UK10K was part of that, but also the OT2D project was published recently, GoNL from the Netherlands, big set of Finns, various different data sets, some of them disease-oriented, like the bipolar and Crohn's disease data sets, and some of them more national. So there's about 35,000 individuals, or 32,000 in the end. These are all lowish coverage. We now, of course, are obtaining tens of thousands of sequences that are high coverage. And we're now beginning to build a new version of this, which we think will have 100,000 individuals. That's around 200,000 haplotypes that will include big components from Africa and or African ancestry and other ancestries, Asian ancestries. So this is the key team of people: Shane McCarthy, Santi in Michigan, so Shane Sanger and Winnie from Oxford. Combined data from 15 data sets. It was non-trivial to get people to pool together information. We recalled everything. We actually think our calls are better than the 1,000 genomes on the 1,000 genomes data set. We have good reasons, at least at SNP sites. We ended up dropping indels, which is unfortunate, and that's what we want to put back into the next version. We intentionally called only at things that were 10 to the minus 4 or above. So that's pretty good, but that's things that are seen 5 times or more. Those are the things which will impute, where we get some sharing and there's some shared information. So singletons, you actually can't say that much about because they're only one individual. So we extracted genotype likelihoods at each of our centers and used a new version of GL phase that Oxford developed, Winnie that was parallelized, and ShapeIt2. So we can't then redistribute this because we've got data from all these different things under different consent structures and so on. They allow this to be pooled. There's no phenotype data, it's just genetic data. But what we can do is make servers available. And I think this is an interesting trend. So there's an imputation service at Sanger and at Michigan. There's also a phasing service, which is sort of at Oxford. And there's over, these are out-of-date numbers. So there are, I think, probably over 5 million, 8 million genomes have now been imputed on these. Now, we're working on a basis of doing that through secure gateways, and we don't keep any information from that except for who sent in how many samples, sort of accounting information. So probably the same things have been imputed more than once, at least in some cases. But still, I find it kind of remarkable that there are clearly many millions of genotype samples out there in the world that have been imputed. In some ways, why bother to download all this data, run these rather complex programs, parallelize them, and so on? It's a heavy compute operation. So these are very streamlined and standardized, and people can be assured that everything will be done the same way. And then if you integrate things, that things will be done consistently. So this is interesting because we've been through swings in the informatics community of doing things locally and remotely. Like with database searching, used to be in the 80s done locally, and then we all switched to blast servers, and then people now, do reference matching locally, and so on. And I think having well-known services which are trusted is an interesting way to go for interpretation of data sets that can be maintained. So here's the improvement. Basically, this is the non-reference allele frequency on the x-axis on a logarithmic scale. So this is 0.01, 0.11, 10. This is the 1,000 Genomes Project curve. It's easier to impute common variants. So basically, 1,000 genomes can impute things from 5% or from 1% upwards reasonably well. This is if you add UK 10K. This is adding the first version of the HRC. This is a rephased version. So these look, there's clearly a difference here, but it's quite a big difference. We're now, at 0.0, well, at this point here, we're doing effectively about 10 times as well as we were doing at 10 times that before. So this sort of factor of 10 is a rule of thumb for low frequency variants. We're going tenfold deeper into rare allele frequencies in the HRC. So what's one of the things that's behind that, at least the Sanger server, is this new data structure, which was mentioned, this PBWT for haplotype matching, where essentially we've taken, if you take a whole set of sequences here represented as zeros and ones, and you sort them and look at what the next position, what the next base value is. So these are sorted running backwards. This says 0, 0, 1, lots of zeros, 1, and so and the red things are what each one shares with the one above. So if you just look at how this left-hand sorted prefix predicts the next value, that column of the next value I call the PBW, the positional Burrows-Wheeler transform at that site. And that's the same essential data structure, and we'll come back to this, as the Burrows-Wheeler transform. So matches are adjacent in the sort order because they're sorted, so things that are identical or for some region. These are maximal matches within the data set. And you can use all the same algorithms that are used in BWA and Bowtie and so on for haplotype matching as there you did for sequence matching. Okay. And one thing that happens is that the values in this column are highly correlated because the context is predictive. So if these two sequences are the same all the way along here, then it's very likely that they're the same at the next position, and they are. So they don't look that, there's a clump of ones here and a clump of zeros here. They don't look that clumped. But when you get 10,000 objects in this, then you get huge long strings of ones and zeros. They're very clumped. And it's that clumping that makes it predictive. It means that you can predict the next value, and that's supportive. But it also allows you to compress it, because you can run length encode these things. So here we have data compression. We can run length encode the columns. This is going up from 1,000 samples to 100,000 samples. This is simulated data. And we're going up from 4.6 bytes per site to 20 bytes per site on a 100-fold increase. So 5-fold increase in data set size for a 100-fold increase in the amount of data, of raw data. So of course, there are more rare variants here. It's a bit simpler to, but the gain over gzip is also going up quite dramatically. So we've got a big gain over gzip. So really, we're using the genetic structure to help compress. And that carries across to the 1,000 genomes data. We get a 6-fold factor over gzip, and we get a 12-fold factor for the HRC over gzip. So these are order of magnitude improvements. So Hung Lee's BGT package, which is on his GitHub site, is another implementation of the same ideas. And in fact, there are beginning to be other ones as well. So that's compression. We can also match very fast. So if you do naive matching, which is not totally naive, using some sort of extend process, then you have linear performance. This is going from 1,000 to 50,000, and this is 50 times as much time as this. So these are in seconds, I think, to match 1,000 new chromosomal sequences. If we build a full index, like an FM index, then just as the BWA is independent of database size, this is essentially independent. We're taking a second up to 20,000. And then what happened here is that I started swapping on my laptop because the index got too big. But you can also have intermediate things that use the same data structure that are fully maintained in memory. And so there's a whole set of algorithms that are very cost efficient, hundreds of times. OK, so here we've got a compressed data structure, and we can do that on the compressed data structure. We've got a compressed data structure where we have fast matching algorithms, which is what we said we wanted at the beginning. We can use this for imputation because we're matching. We now take a new individual and we say, where do they match? Which people in the whole data set do they match to best? And then at the sites that are genotyped, and then you can copy down the information from those. And if we pick up the 20 or so best matching individuals, we can copy from them. And that's much faster, essentially going from 55 seconds to 40 minutes if it's pre-phased, which is the direct comparison with impute. 87 megabytes to 8 gigabytes, so of the order of 1 to 2 orders of magnitude faster, getting closer to 2. There's a slight degradation at the moment in performance, so this is against comparing Minimac to PBWT, but we're still a lot better than the 1,000 Genomes project. So it's very fast imputation, and that's what's behind the Sanger impute server that allows us to do millions of genomes. We can also use that for phasing, as we heard, and in fact, Poru Lo kind of has led, I think, the most successful implementation of this so far. This is in press in Nature Genetics, and I should have said that the HRC paper was recently published, I think a week or two ago in Nature Genetics, announcing the resource, and the Michigan Impute server also published. So what Poru does is using Burrows-Wheeler transform to build a whole set of little trees of possible haplotype extensions. So these are like the trees that you get in Beagle that it uses for representing things. And it builds one at each het position, actually, for a sample. And because the data structure is all pre-sorted, that's a very fast thing to do. It's, in fact, linear time to build this set of structures. And then what he does is extend out an ensemble of likely diplotypes in a sort of particle filtering method. And he calculates likelihoods in, again, order m time. M is the size of the data set. And that's very fast and accurate. So here we have ShapeIt2. Here's ShapeIt2 in its mode. This is reference panel size from 15 to 50,000. This is CPU time per target genome. These are two versions of Eagle, essentially flat. The first version of this was ShapeIt2 with no MCMC, which is the blue line. So you can be fast with ShapeIt, but if you don't use MCMC, then you're fast but not so accurate. If you use MCMC, then you slow down and you're more accurate. ShapeIt1 was worse than, Eagle 1 was worse than ShapeIt. So that's why Eagle 2 was developed, so as to sort of be uniformly better. And I think achieves that. OK. And that's now used to phase the samples that are sent to the Sanger server. And maybe we even have a straight phasing option. Yeah. So, okay, so that's using those things. And let's see. And I wanted to draw attention to this paper, which I'm not involved in, but Benedict Payton and the VG team, Eric is there, and Adam Novak in particular, have realized that you could apply the PBWT to the graph extension and merge the two ideas I mentioned. And that's in Algorithms in Bioinformatics, just came out last week. And actually, my view is that this is the BWT, the Burrows-Wheeler on the sequence of node identifiers. And that's actually what we're doing with my positional one. So that there's a very clean relationship between all the kind of computer science theories on Burrows-Wheeler transforms and these equivalent things on paths through graphs. So we can get efficient ways to compress lots of different versions of the human genome running through a graph and use that, I believe, for interpretation. So I wanted to talk about one more thing, which is the Read Server project, which has been led initially by Shane and more recently by Thomas Keane and colleagues, Dirk and Ji Cheng. And the idea here is, how far can we go in building in all the knowledge that we've seen before? The ultimate thing is to take all the reads that we've seen before, and say, what have we seen? Can we build a queryable data structure that's feasible on really large amounts of read data, which will be a reference-free analysis in some sense, or the reference being this read set. So we thought we'd try and build something that allows queries over the complete set of 1,000 genomes reads, which samples contain a given sequence, what reads support a sequence. Could you do local assembly across all the samples, seeded by some match, and say what sequence have we ever seen that around this, and how does it relate to itself? Can you find new strings in there which aren't in any reference or variant database and define new variants? So they built a full-text searchable index of all the reads that can sit in memory. And it provides a rapid return of all the reads containing an arbitrary k-mer. And a secondary index then tells you which samples each read is in. So this is the 2,500 samples. There's about 10 to the 12 reads, 87 terabases. We need to error correct them. To compress them, you have to get rid of errors, basically. And then what happens is that actually each read that is in the reference or in commonly seen, the actual read itself, that string of 100 bases, will have been seen many times. And so you can make that unique, take the unique set of reads, and that reduces by error correction, and then taking a unique set down to 5 terabases. So that's a major 15 to 20-fold compression. And then that 5 terabases, using the Burrows-Wheeler, because there's still lots of shared information there are lots of off-by-one structures, can be compressed down to about half a terabyte, which is holdable in memory, very small fraction, under 1% or 0.01 bytes per raw base pair. And the ways that you do error correction and compression matter, and I think we're not at the end of that. There's an implementation that Ji Cheng built with a whole set of sort of distributed servers. You can now query with a 30-mer or a 70-mer. You can find how many matches you get, how many unique read sequences you have, and then you can pull those out at about 5 microseconds per read. So this is really something you could use as a server. There's an implementation available on GitHub called the Read Server. It's with a toy metagenomics data set. You can download and use that. So one thing we did is compare GRCH37, that's this oval, GRCH38, all the variants called in 1,000 genomes, and everything that's in our read set. And firstly, there's a lot of things shared by the references that we see, 2.5 gigs about. there's about another large amount, 2.5 billion or 2 and a bit billion 31mers, which are found in the 1,000 genomes variants, which we find, but are not in the references. About as much, actually, as the reference itself at the 31mer level. There's a very big number here of things only seen in the read server, probably dominated, or actually almost certainly dominated by errors. So there's where we could get more compression if we could find those. So we were somewhat conservative about what we removed. And then there are other numbers in other places that are interesting, but you'll have to read the paper to look at those. So the one thing that's a nice illustration of this is you don't just have to look now for things that are in the reference at all. And here we looked at all the virus sequence in GenBank, and we compared it to all the samples, and that could be done in under 2 days because of the speed of the database of the server. And you find some things like adenovirus and CMV, and we found 6 samples containing HTLV1, which is a virus that's known to have originated in West Africa. So this is from a previous paper about HTLV1. It got into the Caribbean and South America and also into East Asia. And we found it in a number of samples which were from blood, originally in the 1,000 Genomes Project, both from West Africa and from South America, which makes sense that it's circulating in those populations. So that's a tie-in there. But there are other things that you can look for, and one can use this in all sorts of interesting ways. So I'm just going to finish off by saying I was at a meeting. I talked about this at ECCB. For some of you might have been there. There was a meeting in Dargstuhl. That's the German computer science kind of retreat workshop center in West Germany at the beginning of September. And we were asking, could we imagine a Google-like search on all the world's sequence? What would it do? Let's say that we really could get distributed access. Could we extend out this read server idea? So you could search with a 50mer and get a set of link outs with further information. Now, you wouldn't want all the samples that contained it, because anything in the reference would be contained in too many. You sort of want a hierarchy, a bit like PageRank does anyway. You'd want to know if it's in a reference genome, then where it is and what the gene is. If it's in a known variant that people have seen before, its name and frequency and links to the standard resources for those things. If it's a known splice variant, you'd like to know what transcript it is. So you can imagine there's 2 parts. One is actually finding the matches and building the index, and the other is working out what information is most useful. But I think that there's a sort of interesting concept here. And I think it's actually quite like the way people used to use BLAST servers. They used to, when in the early 90s or before we had reference genomes, people would struggle to pull out their gene sequence. And then the first thing they'd do is go and look for matches. And they'd get these hits and sift through. Some of them would be boring and some interesting. And people tried to rank the hits to be the most informative. Is it in a PFAM domain or something, seeing Alex in the front row? So how could you build it? Well, there's a lot known in the computer science community, actually, about how to build such things. You crawl across public data sets, extract digests of relevant information for central indexing. We would need to error correct. It's a bit like you can't hold it all centrally. You need to hold condensed digest information, which is sufficient to build a search tool. And then you'd want a replicated index that would give you rapid responses. And there are different discussions. So one could imagine this just on public data, or even one could imagine people allowing you to search, return a hit, and say you have to kind of get more authentication to get in. So the different versions of this. But I think we should be bold in thinking about the way people will interact with sequence data globally and use some of the infrastructure that people have thought about. So I'm going to finish there. I wanted to, what's this? I see. This is the Global Alliance. I think, give a quick plug. The VG project is sitting within the Global Alliance coordination. Now BAM, CRAM, and VCF are also there. There's a strong effort to provide a server streaming client, streaming server client implementation so that one can access things through web interfaces remotely, more cleanly. This is trying to coordinate the use of genomic data. There's lots of working groups, the API and the file formats and so on, but there's also regulatory and ethics and security and clinical working groups. And I'll finish there, and these are links to various things I've talked about. Many of them have things in the archive or publications, and thanks very much.
Where's right at the back there? While the microphone's running up, I wanted to raise a question about the use of this graph genome approach. I mean, one of the things I think actually we were just having a discussion on Twitter was how long the community took to switch from build 37 to build 38. It's been over 2 years, and there's some resources that have changed. So obviously, there's a lot of inertia in this system, and the challenge with the challenge with graph genomes is you're not just switching to a new reference, you're switching to a fundamentally, conceptually different way of thinking about things. So what are the key resources that we need to make sure we have in place so that people can actually understand and move to this graph genome world? For instance, what does a genome browser really look like in a graph-based world, and how do people interact with that data in a way that they fully understand?
Well, I think we're going to want browsers. We're going to want the toolkit around, but we shouldn't not be building the toolkit just because it seems daunting.
Of course.
And it has to be demonstrably better. I think 38 is demonstrably better than 37. And I think we have to produce a toolkit and show that you can, if you use it, that can help handle information better, more accurately, or faster, or more compressed, or all three. And what will they look like? I think there are ideas, and there will be more ideas. I'm not going to really go into.
Right, that sounds
I think you're going to want to see it from a more individualized point of view,
sure.
basically. And that's a direction of travel for the browsers, is to be able to look at data from an individualized point of view.
Sure. Excellent. Thanks, Richard. Question at the back there?
Yeah. I have a question that comes back to the VG project. When you do a variant coding like stuff, you say that you add a variant in the graph, but just in a temporary manner. And I wonder why you add this variant not definitely in the graph, and what is the threshold that you choose for why I add this just for the comparison of why I don't add this definitely in the graph.
Yeah, so I understand, and one could say that about the current reference. Let's say somebody has a lot of data, all of which has 2 Gs at this location and not 1 G. Why shouldn't you change the reference to reflect what you see? But of course, then all the other things that you know about that reference, you lose instantly. And probably you don't really want to do that. And I think you can have it's conceivable. There's nothing technically to stop you accumulating things. But I think your kind of confidence, there's a lot of care taken by the GRC. It's good to see when they make changes, they're careful about that. And they're more careful even than the people who are, they're clearly more careful, I should say, than people making variant calls, although those people are also being careful. So I think it's just pragmatic. I think in practice what we'll do is build reference graphs which capture a lot, which we can build some infrastructure around, some resources around, and people can use those for a bit. And there might be new versions. I mean, it's early days, and exactly how you work, people might work inside a group slightly differently. That's my current view.
So that was great, and I think the graph stuff is really good, and it's sort of a follow-up to Dan's question, because it looked like from the data that you were showing that most of those alignment and variant calling comparisons were on graphs of just individual regions. Is that correct?
Well, that's the evaluation that's been taking place, led by Benedict, has been focused on these regions. Because, frankly, it started before the machinery was really in place to work genome-wide. I mean, these things are going in parallel. So this is something in process, but I think now, actually, we are at a stage where one can start doing whole human genome
Okay,
things.
that was my question, because I know that has been my perception of one of the challenges of this is having the scale to doing the whole human genome.
Yeah, so a definite goal of VG is to put yourself where you can do that, and I think we're about there, but yeah. And certainly we've been testing on yeast, you can run through all sorts of things. That's also 12 megabases though, so, but then we can rebuild on human and do things in days.
We had one question here, Michael first, and then.
Yeah, so I'm very excited about our glorious graph genome future. Mm-hmm. I'm curious as to what thoughts you have on how this will affect the analysis of count assays like RNA-seq, ChIP-seq. How will you do this in a graph genome background?
Mm-hmm. Well, that's fine. You'll map all that stuff, and that's an important thing also. They'll map to paths. You can look at coverage of nodes and sequence. Some of that will be in the reference. Some might be in the current reference. So you're going to have to say that there are variants from the reference which have this degree of coverage. If you're interested in it on a sort of current reference coordinate system, the translation can place that coverage. And our view would be that it will improve the ability to map honestly and sort of to reduce the issues created by mapping errors. But you can also look for support for, so I think it would be a much better framework for allele-specific mapping, for example. One's neutral, you'll avoid reference bias in allele-specific mapping, and you can ask questions about specific alleles and their coverage, and there'll be a global depth map on the graph, essentially. And how it gets reported depends on how much effort you want to go into.
We're slightly over time, so I think we have time for one quick question, and then.
So you've been describing the variant graph for normal genomes, and you can put as many inverted commas around normal as you want. So how about clinical applications? And if you're thinking in terms of cancer, it would be possible to create a reference graph for, say, melanoma, for leukemia, and so on. Are there people working on that already?
Well, there are other sorts of graphs which are not entirely independent. So one are what has happened to a cancer, particularly with lots of breakage, and you can represent its relationship to the reference as tying together bits of the reference and repeating them and so on, and maybe bits of novel sequence. And actually, I think having the machinery to handle these graph structures genome-wide, so one will need specific tools to build those things, but having the machinery to handle them and look at them, represent them, will really help that way of looking at cancer. Whether you get enough information to give you a completely accurate picture isn't clear. There are interesting questions about that. And that is related to the other use, which is assembly. And I think assembly graphs, so graphs have come up historically in assembly for the last 20 years. The same data structures, there's also relationships there. And having the ability to map reads, for example, into graphs allows us to, instead of just taking the context from an assembly, we can now map reads into the assembly graph. And I think there are relationships for species where we don't have such a good reference as we do. We will use assembly graphs as reference graphs. It'll enable us to do that. So, yeah.